Hi everyone, this is Kumar, Associate Professor, SNS College of Pharmacy and Health Sciences, belongs to Pharmaceutics Department. So like the previous discussion, we are going to discuss about the one more advanced CRISPR CAS system. It's nothing but a CRISPR CAS 13. It's a very recently innovated during the year of 2016-2017. It's a class 2 type 6 CRISPR system. It's a RNA-guided RNA systems. When compared to the previous two systems, it's slightly differentiated. It only cuts or targets the single-stranded RNA and not DNA. So there is a reason why it plays a vital role in the RNA diagnosis as well as the RNA therapeutics. So what are the key variants here? There are typically three variants that performs the genome editing as well as the diagnostic and therapeutic applications. They are CAS 13A, one enzyme, CAS 13B as well as the CAS 13D. Next, the advantage of the CAS 13 is here there is no PAM recognition site required. So it only need a protospacer flanking sequence with the PFS in the same subtypes. It performs the editing, cutting as well as the removal things. The previous CAS 9 as well as the CAS 12A, they requires a PAM recognition site. There is one of the big advantage for this system. That is the reason why it performs in the RNA systems. So what is the unique? It exactly cleavage the nearby non-target single-stranded RNA upon activation. So once the CAS 13 system activates, it's exactly identify the nearby non-target single-stranded RNA and makes the changes. So it enables and performs for the diagnostic and transient knockdown system. Next, the same like previous, what it plays the natural role in bacteria. The same like it plays a vital role of the bacteria when compared to the molecular basis category. So it plays adaptive immune defense in bacteria archaea against especially RNA based viruses and RNA based plasmids and bacteriophages. Next, the same like DNA, how it's stored in the CRISPR array. Here RNA captured from the virus genome and it's stored as a spacers. Next, when it binds or target with the single strand RNA, it performs the specific cleavage and thereby it prevents the integration of the viral genome as well as the bacterial genome. So where it exactly prevent the integration of viral genome versus the bacterial genome? So it prevents the viral genome integration with the bacterial genome in its transcription site, especially RNA multiplication. Thereby it protects cell. Here there is no DNA involvement, only at the translation site only. Next, what is the mechanism? It performs direct repair plus spacer. Thereby it uses the tracer RNA like a basic elements to identify the CRISPR RNA based RNA editing as well as the knockout mechanism. Next, it targets complementary single strand RNA. Again I am stressing it's only RNA, not a single strand DNA. Here there is no double strand DNA unwinding needed because it only targets or fixes its mechanism based on the RNA basis. Next, it exactly performs or cleave the target RNA at the specific site. Next, it indiscriminates the single-stranded RNA after cleavage in a particular area with the help of the collateral RNase activity. Next, CAS13 used binding only applications like imaging, splicing modulations. That also one of the mechanism. So what are the editing and functional outcomes of these CAS13A systems? Here there is no permanent genome changes. That is the main advantages. Next thing is the same like the previous knockdown at the mRNA level. It cleaves the exact target mRNA. Thereby it prevents or reduces the gene expression by alternating the NRA to the small interference RNA. Next, RNA editing. It fused ADR pathway to precise base editing without the double strand breaks. Next, collateral cleavage. It is an ultra-sensitive detection of RNA targets. Example, viral RNA and mutation. So that is the reason it performs an excellent role in the after transcription to the translation area. Next, what are the applications? First, diagnostic. It performs excellent diagnostic under the Sherlock platform. So what are the things it performs under the diagnostic? Like rapid and portable RNA detection. Examples, viruses like SARS, Zika and cancer mutations, and of course SNPs, it's nothing but a single nucleotide polymorphisms. So in therapeutics, what are the main role? To identify the toxic RNAs and perform knockdown. Knockdown is nothing but to remove the toxic RNA, thereby the toxic RNA's expression prevented. To identify the RNA viruses, to knock down the RNA viruses, to knock down the cancer oncogenes. Likewise, there are so many vital roles it plays. Next, RNA editing for diseases. Example, MCP2 duplication syndrome. So MCP duplication syndrome is nothing but it is a gene, MECP2, it is a gene, methyl CpG binding protein 2. It's a crucial gene located in the X chromosome and it produces a important protein essential for the normal nerve cell function, that is nothing but a neuron development. Next, what are the research it performs? Live cell RNA imaging, RNA splicing and modulation of RNA, and plays vital role in the transcriptomics and antiviral strategies. So advantages, when compared to the previous CAS9 and CAS12A systems, what are the advantages? It's a RNA specific, there is no genomic edit, it's a multiplexable, and the size of delivery is very smaller when compared to the previous CAS systems. So what are the advances in the 2026 under these CAS13A systems? It is a high fidelity in nature, so almost it minimizes the errors. Next, it's a dual guide for SNP specificity and in vivo RNA therapies.